Hello and welcome to the Bloodstream Podcast, a show serving the greater bleeding disorders community. Brought to you by Believe Limited and Bloodstream Media and made possible thanks to our presenting sponsor, Takeda. I am your patient advocate and host, Patrick James Lynch.
And I'm your healthcare advocate, nonprofit nerd, and your other host, Amy Board, reminding you to please speak with a healthcare professional before making any treatment decisions.
On today's show, Craig Benson from Sanofi, the head of clinical research, executive head of clinical research. It's a very fancy title, but basically he oversees clinical research.
Cool.
He's coming on to talk new treatments.
Cool.
And we have the newest gene therapy segment with a focus on the psychological impact of gene therapy. We'll hear from Chip Haley, who lives with hemophilia B, and Simon Fletcher, a nurse and PhD candidate who helps break things down from a clinical and compassionate lens. We have got all of that and more on this cool episode of Bloodstream. Welcome to Bloodstream.
This is so cool. It's so cool. Listeners, as always, thank you for joining us today. And if you like what you hear, subscribe to this episode and tell all of your friends on the social media. We would love that. We want all of the listeners, all of them.
Amy has said cool 15 times as though it's not currently 98 degrees outside.
I know.
Just slaughtering the word cool when it couldn't be less true about the present circumstances. Listeners, I also want to let you know that the Bloodstream Podcast is indeed made possible thanks to our presenting sponsor, Takeda. Yes, that's right, Takeda. Takeda's got this website, bleedingdisorders.com, where you can learn all about Takeda's resources for and commitment to the bleeding disorders community. Takeda, Amy, believes in a world free of bleeds.
Which is cool.
And they're dedicated more than ever in their efforts to offer a wide range of programs and support to help patients on their treatment journey, wherever on that journey they may be, and you can learn more by simply visiting bleedingdisorders.com. One more time for the folks in the back, bleedingdisorders.com. And for their founding and ongoing support of the Bloodstream Podcast, the #1 hemophilia podcast, according to FeedSpot. Yes. I would just like to say, thanks, Takeda.
Thank you, Takeda. We should bring that up. We're the #1 bleeding disorders podcast on FeedSpot.
True.
And there's some others on there that are legit, so it's kind of cool.
It is kind of cool. So shout out FeedSpot. Appreciate the love. Amy Board got a lot as always, but I want to let you know yesterday I popped down to Washington, D.C. I'm in New York. I did a little pop down and met with some people during the International Society of Thrombosis and Hemostasis' annual meeting, or ISTH.
ISTH. And that meeting typically is overseas, right? It's like in Europe or something. And this year it happens to be in Washington, D.C.?
Yeah, I think it goes like Europe or the States, then somewhere like Persian Gulf, Middle East, and then somewhere like Asia Pacific. It kind of jumps around. I don't remember the last time it was in the States. I'm sure it has been in the last 15 years. But yeah, enough people were like, are you going? And when I realized I'm close enough in New York, I can at least pop down and see some people. One of the people that I bumped into was none other than NBDF COO Dawn Rodolini, which meant I got to tell her, and I'm going to tell you right now, Amy Board.
Yes.
Dismissed.
I know. I'm so glad you brought it up. Yes.
Dismissed. Guys, Dismissed is a really good film, and it doesn't even have music yet. It's not even done. And I'm telling you, it's gonna make an impact at this year's BDC.
So y'all, Believe and Sanofi have partnered once again to bring a feature-length documentary to BDC and to the community this year. In the past, we've done Let's Talk, which was a look at mental health in our community. We've done
On the Shoulders of Giants.
And then we did Read and Fighting Impossible. And this year, Dismissed is about women with hemophilia. And the response is already buzzing. Hannah Berry, who works here in patient advocacy, has already gotten so many phone calls and texts from chapter leaders that want it at their events, which has never happened before. A couple chapters asked if they could get it before BDC, and we had to be like, absolutely not. It's not even finished. But anyway, Patrick saw it, and
I did.
Anyway, yeah.
I was excited to see it, knowing that I was going to be bumping into some people from Sanofi while I was down there. I wanted to be able to speak to it specifically. And then when I saw Dawn, I was like, I was just staring at your face, and guess what? It's really good.
I know,
so
it's
Dawn,
so good.
Dawn is in the film.
Dawn is in the film, and Dawn is like a through line of the film. She's marvelous. And if you're going to BDC this year, you have to check it out. Schedule it on your calendars. It's Friday, August 22nd is going to be the premiere. The premiere is going to be outside, even in Denver. It's going to be glorious. 8 p.m. And then if you're not going to BDC, it will be free on the website so you can watch it, or check out your local chapter events, because so many chapters are going to screen the film for their communities, their local communities. It's a really cool film. I'm getting goosebumps just thinking about it. So thanks for bringing it up, PJL.
My pleasure. My pleasure. So, all right, that's coming soon. More on Dismissed as we approach the BDC, that is for sure. And now this next segment is brought to you by Genentech. There's a new short film out right now. This one you don't have to wait for. This one you can go watch right now. And it's dedicated to moms who care for their sons with hemophilia A. It's an honest and visually striking journey about a mother going through the emotional whirlwind of raising a son with hemophilia A. The story is inspired by thousands of real moms and serves as a thank you for all that you do to make the hemophilia A caregiver community a place of love and resilience. Visit www.hemashort.com to watch the short film. Again, that's www.hemashort.com. Okay, so back in 2019, Spark Therapeutics became a member of the Roche Group. Roche is the umbrella company that Genentech sits under, that many of us here in the United States are familiar. Well, earlier this year, in January, Roche announced that Spark will become fully integrated into the Roche Group, and as a result, in the US going forward, Genentech will be the point of contact for the hemophilia A gene therapy program. So that's bullet number one. This has been in the works for the last 5-6 years. So while it may sound like, wait, where did this come from? No, Spark's actually been a part of Roche since 2019, even though it's been continuing to operate independently. But now it is being fully integrated, fully folded into the Roche Group. And in the United States, that means its gene therapy hemophilia A program, the patient advocacy relations function, that is now going to drive through Genentech. So that's point number one. Point number two is that the clinical trial program that was previously with Spark Therapeutics and paused back in December of 2024 is going to be resuming kind of under Genentech, but in a new capacity. So what are they calling it? So there is an enhanced function factor 8 variant that is now being introduced into a new phase 2B program. They stopped phase 3 of the initial gene therapy program before anybody was dosed, and they decided, not due to safety concerns, but due to a commitment to creating a one-time gene therapy that improves hemostasis and lowers treatment burden, they discovered that this variant might be the way to go. So there's been a transition, and now, what are they calling it? The next generation program. So what is now going to be taking place is a phase 2B of what they're calling their next generation hemophilia A gene therapy program via Genentech. So 2 points here. Spark is going to be transitioned out. There will be no more Spark in due time. It's all going to be running through Genentech. And the Spark gene therapy clinical trial program, which did stop at the end of last year before initiating phase 3, I guess it actually has been discontinued, would be the appropriate word to use. And now this new variant's been introduced. The trial is now being reimagined as a next generation program. Phase 2B will commence. And Amy, hopefully the result of this will be an efficacious, durable, one-time gene therapy. That is the goal after all.
Honestly, thank you for bringing this to my attention. I feel like there's a bunch of misinformation out there about that Spark trial and what really happened there. And I love that the science is continuing to move forward and it's new. That it feels like there's something new. So for hemophilia A community members out there, this is something to keep an eye on about how this science is going. And does it say in this letter, Patrick, where they are, what phase they're at with this clinical trial? Are they in phase 1 or phase 2?
Phase 2B. So I don't understand the details behind this. I don't know exactly why it is that because this variant has been studied elsewhere, Genentech was able to essentially expedite this next generation program doesn't have to start at phase 1 all over again, kind
Okay.
of gets to start, a little bit down the road. Why exactly? I don't know, but it is notable because that is not the most common, from my perspective,
Right.
that's not very common. So
Okay.
that is notable, certainly something to follow up on. And as you referenced, Amy, if I didn't mention it, the information that I was just presenting was published in a letter from Spark and Genentech dated May 27th of 2025. We'll put a link in the program notes, so if you want to review it on your own, you can certainly do that. But yeah, we're going to be saying goodbye to Spark. We're going to be saying goodbye to some of the people at Spark who will go on to do whatever they're going to do, but they won't necessarily be a part of this community anymore. So it's definitely a transition and something that, as Amy said, we'll be tracking as things unfold.
Cool. Thank you. Thank you, PGL, for bringing that to all of our attention.
Amy,
Patrick.
you want to play a game?
I don't know. Yes, I do. I just want listeners to know that I don't know what's happening.
So I played a game that Amy's now going to play. It's a 3-part game, and I want
Okay.
to see if you can do better than I did.
Okay.
Okay?
Okay.
Qfitlia.
Qfitlia.
What's the derivation of the name Qfitlia?
I don't know what that means.
Where does that name come from? So whenever they make these drugs, their commercial name is always, the syllables are always like, oh, this is coming from here, this comes from over there, that comes from Qfitlia. Where does it come from?
I don't know. I like a huge board meeting of people, and there's the 26 letters of the alphabet, and everyone has to vote which letter that it starts. And it's just like a bunch of people in a room, and they've got like coffee and soft drinks in there. And like Q was the one the most, and then they like were like, okay, let's come up with Q names.
So Q is for sub-Q, but I like what you said.
Okay.
Now, fit. Right there in the middle. Fit. Where's the fit come from?
Fitusiran, right?
Bingo,
Okay.
Amy Board. Well
Okay.
done. Okay.
Okay.
You're on the board, Bordeaux. You're on the
Okay.
board.
Okay.
And Elia.
That's got to be like
Qfitlia.
like a
Leah.
mab, like, like Sizamab is like a certain type of thing. It's this like Ilia's got to be like a
Where do we say things that end in Elia?
hemophilia.
Boom, Bordeaux. Well done.
Okay.
Hey, you got 2 out of 3.
Okay.
And frankly, your answer for Q is, I'll give you 3 for 3, actually.
I'm actually going to talk to some of our pharmaceutical pals and be like, if you're wondering about how to come up with names, here's a process.
As VP of Engagement and Programs, do I have
With
a program?
BFA in musical theater.
Well, good news is we have people who have expertise beyond BFAs that can talk to us
Yes.
more about Qfitlia and other stuff as well. So, little background here. Qfitlia is a recently available new treatment option for people with hemophilia. I knew it was on my radar, previously known as Fitusiran, but to be honest, I didn't know a ton about it. So I reached out to someone at Sanofi to say, could we get a representative to come and share with the audience more about this? And Craig Benson, who is their head of clinical research, was the person who was identified. He's fantastic. I really enjoyed talking with him. I find the science quite compelling. So you decide how you feel about it. Here's Craig Benson talking about Qfitlia and new treatments. So joining me now is Craig Benson. Craig, what is your title exactly at Sanofi?
I am the executive global project head of our hemophilia cluster in rare disease clinical development.
I like the use of cluster in that. You don't get a lot of use of cluster in a title description.
Thank you. Signifies that I oversee multiple clinical development programs, and at Sanofi we have a number of ongoing, clinical development programs in hemophilia. So usually it's project based, but I think we're pretty fortunate at Sanofi. We have a number of programs and projects to advance the science for hemophilia.
And today we're in particular talking about Qfitlia. Probably to some degree we can't help but talk a little about Altuvio because it still is relatively new to the market, so to speak, as well. But before getting into those specifically, just for additional background, Craig, how long have you been working at Sanofi in this capacity or related capacities?
Great question. I've been overseeing our clinical development programs in hemophilia for 3 years, but was previously on Altuviiio, leading that program specifically, and at Sanofi now for nearly 8 years, all in hemophilia.
Got it.
Yeah.
Okay. And we hear from people who come into hemophilia how it's unique, the ways its tentacles can get into people. I'm curious though, what has your experience been compared to other stints you've had in your professional career?
Yeah, it's been. I'm actually a cardiologist by clinical training, and so if you would have told me when I finished medical school that I'd go on,
Hemophilia, naturally.
go on to work in advancing new therapies for people living with hemophilia, I'd say you're crazy. But I actually started off in clinical development at Biogen/BioVerative on Altuviiio, and had an opportunity to join Sanofi and to work on fitusiran. And then, obviously there was a merger and acquisition. Sanofi acquired Altuviiio. But I remember when I first joined, one of the very first tasks my manager gave me at that time was to lead a patient advisory panel on our first in human phase 1 study for Altuviiio. And I remember it was day long here in Boston, and I was just, I walked out. When you lead those types of, day long meetings, it's exhausting, but I remember being so inspired by hearing and working with the patients and people living with hemophilia, hearing their stories about what they have to deal with, their pain, how they have to treat, the number of injections they have to give. And it was really, for me, it just ignited a real passion to continue to move the science forward. And here we are, 8-9 years later now. A lot of people, I'll just highlight that, it takes a village. It really does. I am a spokesperson today for so many people that have worked so tirelessly with a lot of support to bring these, what may seem like crazy ideas, to move them forward, to always keep the patient head up.
So let's talk about the crazy idea that became Altuviiio first, actually, because given what you just shared about your background and the timeline, let's start chronologically.
Yeah.
Altuviiio, help listeners who may not be as familiar understand what exactly is it, how does it work, in what ways is it novel, and in what ways is it quite precedented?
Yeah, great. So Altuviiio, the kind of the brainchild of Altuviiio, occurred right as, and maybe even just before Eloctate was approved. So Altuviiio is a factor VIII replacement therapy for hemophilia A. And, there was, I think, tremendous excitement from the community when Eloctate and Alprolix were approved and hit the market first, long-acting factor VIII and factor IX therapies for hemophilia A and hemophilia B. However, it was very clear for hemophilia A very early, wait a minute, they're still injecting once, or a couple, 2, sometimes 3 times a week, and their protection level is dropping down to levels that put them still at severe risk for bleeding, spontaneous bleeding. And so it was realized very quickly, okay, well, why is that? Factor VIII binds to this very large protein in our blood called von Willebrand. And it's a blessing and a curse. By binding, it kind of keeps it in its blood a little longer, but by doing so, that protein is limited in how long it can stay in the blood, really how long von Willebrand stays in the blood, which is a half-life of about 19 hours. So in order to
just to pause you right there, Craig,
Yeah.
as a patient who my life grew up hearing about my half-life and wondering, oh, it was my half-life. How long do I have to infuse the half-life? Oh, now I have 50%.
Yeah.
Is it fair to say, or is it too oversimplifying to say that historically speaking, it's actually the von Willebrand factor that is bound to factor VIII that has more to do with historically factor VIII's longevity in the bloodstream as an active viable protein?
It's a huge part of it. It's a huge part. I think early factor replacement therapies for hemophilia, factor VIII replacement therapies, maybe didn't even get so long. They weren't able to. The technology wasn't yet mature enough. So Aloctate came along, used the FC technology, which uses a recycling pathway in cells that allows it to stick around a little bit longer. But even that was still not enough, because in blood it binds to a specific location on von Willebrand called the D prime D3 molecular subunit of von Willebrand. Okay, so if that's where factor VIII binds, is there any way, and this is I think the crazy, really exciting idea.
This is where it gets fun.
About Altuvio, right? Where it gets really good is that, okay, if that's where it's binding, is there any way that we can actually like build that right into the molecule? Okay, we've added, the FC component to factor VIII. Can we add any more of these kind of molecular parts to extend the half-life? So that's exactly what our scientists did. And this was all work that was done prior to the BioVeritiv spin-out, which is to actually engineer part of that D prime D3 subunit of von Willebrand directly in to Altuvio. It connects covalently to the factor VIII molecule. And then we had to add some additional X10 linkers that further extend the half-life. They're kind of like streamers on a kite. And this whole construct then is now Altuvio. And that, when tested in humans, we thought and that was the first in human study that I participated in as the early young medical director. And we identified the half-life there to be, nearly double what we were seeing before. And, the phase 3 study of Altuvio read out right in the midst of the, just after the pandemic. And what we observed was that participants could dose once a week. Everybody received the same dose, 50 IU per kg, and which is a little different for the hemophilia community to think about, because it's all been so personalized, right? Personalized. Yeah. But this really kind of changed the factor VIII replacement therapy for people living with hemophilia A. And at the end of that week, your protection level was still above 15% on average. So normal to near normal range for like 4 or 5 days, and then still above, you're still in the mild, almost moderate, in the moderate range of hemophilia.
For sure. And it's funny to think about it in the context.
So, yeah.
It's funny to think about it a little bit just in the context of the gene therapy, sort of like, what's our target for, like, what does efficacy really look like, and what does sustainability really look like?
Yeah.
I remember there was a time where we were talking about 10 and 15% as though it was an acceptable target from a gene therapy perspective. And now here we are talking about that as, the basement, the trough of what you could potentially get on Altuvio. And that's just, again, just to provide a little context, I hear that, and I hear 10 to 15% as a basement. And I think, I remember as a kid growing up, and we were just trying to keep it above 1%. Then I heard about gene therapies targeting 10%. And now we're saying that should be the absolute floor. Just sounds like a great advancement from a therapeutic standpoint.
Yeah, super exciting. And it was great that, at the time there were champions in clinical development, in research at BioVeritiv. Some are still my mentors that really advocated to keep this, to move this forward into the clinic, and very fortunate to see that move forward and get to patients. So,
well, I have more I want to ask you about Altuvio, but I'll ask it a little bit later. I want to switch gears now, and talk about switching gears. So we're treating hemophilia still, but the way in which we're going about it with Qfitlia, completely different. And this is why I wanted to have you on, most especially, Craig. And when we spoke prior to today, the degree to which you helped me understand some of the nuances of Qfitlia, some of the context for Qfitlia, it was, it's an overused expression, but it was pretty eye-opening. And I consider myself educated. I've been following along fitusiran. So I thought, okay, if Craig helped me understand some things about Qfitlia fitusiran, it stands to reason he could help a lot of other people understand too. So that's why I wanted to do this interview today. That was the primary driver. So let me just lay out and let you explain, what is Qfitlia, and big question here, but how does it work?
Yeah, great question. And how much time do you have? We could spend an awful lot of time. But we classify, and I think the community understands that Qfitlia is a rebalancing agent. So within our coagulation system, there are procoagulants. These are molecules that help blood clot, and then there are anticoagulants that float around. These are molecules that prevent blood from clotting, and people living with hemophilia are missing one of the procoagulant molecules, either factor 8 for hemophilia A or factor 9 for hemophilia B. And the way Qfitlia works is there's an imbalance in the coagulation system, which is why people living with hemophilia spontaneously bleed. And so in order to kind of restore that hemostasis, you can replace what's missing, which is factor 8, factor 9 replacement therapies, bypassing agents for those people with inhibitors, try and use it in a slightly different way to restore hemostasis. Well, another way is to rebalance hemostasis by lowering some of the body's own natural anticoagulants to kind of restore that balance. And that's exactly what Qfitlia does. One of the main anticoagulants in all of our bodies is called antithrombin. It's produced in the liver. And so Qfitlia lowers the level of antithrombin in the body, and that allows hemostasis to be restored. And the idea came from there are case reports of people that have hemophilia that also have lower levels of antithrombin. And what those patients experience is far, far, far fewer bleeds than the others. And so that's what Qfitlia does. It restores hemostasis by lowering one of our own anticoagulants called antithrombin. I'll pause there, see if you have any other questions, but there's a lot more to this story because it leverages some really, really cool novel technology.
Well, yeah, let's talk about that. And I assume as part of that, RNA interference is part of the technology you intend to mention. So I find it kind of neat. The science here is pretty cool. So talk a bit about it if you would.
Yeah. So Qfitlia leverages one of our own natural cellular machine processes called RNA-induced silencing complex. The discovery of this pathway was awarded the Nobel Prize back in 1990 by some researchers that are based at the University of Massachusetts. And what it does is it targets mRNA within our cells and naturally degrades them. And so there was, because of this discovery, companies were spun out and developed and started and say, hey, can we leverage this technology, our own natural machinery, to lower RNA in our body for proteins that are maybe too high in certain disease states.
And what's the advantage, Craig, of working on the RNA level as opposed to DNA, protein? How does working on the RNA level have particular benefit from a therapeutic standpoint?
I was just going to say the advantage is that, the RNA is a precursor molecule to the protein, right? Protein is the active functional molecule in the body, and the mRNA, messenger RNA, floats around in the cells before it is ultimately converted to the protein. The benefit of targeting the mRNA level is really durability. So by leveraging this technology, and because antithrombin is primarily produced in the liver, Qfitlia was designed to target lowering antithrombin in the liver. So when you give a dose of it, it's very small, subcutaneous injection, can go right to the liver and knocks down antithrombin levels that last for at least 2 months. So we can dose, Qfitlia is dosed every other month. And that is a big advantage, obviously, for people living with hemophilia, to only have to dose a very small volume subcutaneous injection every other month and achieve and the second thing that's a real advantage is that it provides a very stable knockdown, and that translates to very stable protection levels for people living with hemophilia that are using Qfitlia.
What did you observe in the clinical trials that helped you land on the dosing schedule and this, I mean, once every 2 months? I mean, that's just as a patient, like an extraordinary thing to absorb. Like, wait, what? I could do this 7-minute thing once every 2 months. Talk a
Yes.
little bit about the clinical trial process.
Yeah.
And there were some serious adverse events or SAEs that were handled as part of the clinical trial process. What learnings came out of those? How did that influence? I think that level of transparency would help people understand,
Yeah.
both the value of the trial process and how it came to develop the safety profile that it has today.
Yeah, great. Super important question. And we spent an awful lot of time working on the clinical development and figuring out the right dose for Qfitlia. The currently approved dose for Qfitlia targets a specific antithrombin range. So it's really less about the dose and it's really measuring your antithrombin level. I'll provide a little bit of context. So most people, including people with hemophilia, have an antithrombin level that's around 100. And the what we identified was the range in which we want to target patients is between a level of 15 and 35%. And so that can be achieved with multiple different doses, 50 or 20 every other month, or some rare patients do a little bit more frequently, but by and large, most everybody is on every other month. When we originally designed the phase 3 studies, we were testing a dose that was more frequent and higher. And we identified that led to antithrombin levels for some patients that were persistently less than 10, which resulted for some of those patients in thromboembolic events. And so when we looked at the data, we identified that persistent levels less than 10% were associated with thromboembolic events. And so we then implemented the antithrombin-based dosing regimen based on the clinical data that we observed to target that range of 15 to 35%. And that's what we studied in our long-term extension study, and that data set formed the basis of approval. And what we observed is that our rates of thromboembolic events dropped substantially in using the antithrombin dosing regimen. And we're on par with thromboembolic events for other therapies that people living with hemophilia have to take. So whether, and I think this is one point that maybe is worth mentioning, that, when you restore hemostasis for people with hemophilia, whether it's with factor or non-factor therapies or mimicking agents, those patients have a restored or, near normal level of hemostasis. So they're at now risk for thromboembolic events similar to somebody who maybe doesn't have hemophilia. But if you're older and have high blood pressure and you're a smoker and take long car rides, well, you're probably at risk for, a DVT or thromboembolic events, whether you're, a hemophilia therapy. So when we identified the right dose and tested it, we were very encouraged with the results. That it seems like we have the sweet spot there. We still maintained notable efficacy as well. It's really important to highlight that we saw a 70% reduction in bleeding rates for people on Qfitlia when they target that antithrombin dosing regimen level between 15 and 35%. And the real advantage of targeting antithrombin is that you can measure this. It's not all that different from measuring like a factor level, right? Which is something the community is very comfortable doing. We'll have a lab. You can go to any, lab in the United States and they'll draw blood. You don't have to go to your hemophilia treatment center and you can have your antithrombin level checked.
Well, I mean, you're certainly speaking to my, that resonates with me, because being able to know levels and the inability to do that with certain mimetics continues to be a little bit challenging. And frankly, in a way, almost feels like a little bit of a step backwards. Wait, technology has theoretically advanced here, and in some ways, definitely improvements from what was, but the inability to measure key components, that's, as a patient putting this stuff into your body, a little hard to just take the leap of faith that like, well, we believe it's in this range of, I'd like a little bit more certainty than that. So hearing that we can actually measure the antithrombin levels is quite reassuring. I have a question that's not fully formed, definitely feels ignorant, but feels important to ask. So my understanding is I produce 0 factor VIII. I don't have 2%, I don't have 1%, I have 0%. So when I think about how that disrupts the procoagulant side of the coagulation cascade and the balance from the pro and the anticoagulation sides, no matter what antithrombin may be knocked down to from 100, if it's 35, if it's 15, and I understand we don't want to go lower than 10, I still am producing 0 on the procoagulation side. So how are we achieving balance by lowering on the anticoagulation side if there's still a 0 hanging out on the other end of the scale? Does that make sense?
Yeah, yeah. I think it's a great question, and with any kind of new technology or any drug, quite frankly, finding the right dose is critical, and really what the purpose of a lot of these early, clinical studies are used for. We happen to do it. We've identified some safety signals and revisited that. But that's the purpose, is to find the right line, in terms of safety and efficacy. In other disease areas, you actually test doses until you actually see some safety. And so you don't want to do that, obviously, in a rare disease patient population, especially hemophilia. But it is a balance. And, I wouldn't say it fully, absolutely fully restores hemostasis. All therapies are trying to achieve that. The goal is a cure, right? Let's be clear. All of us want to see, whether you're somebody living with hemophilia or you're in drug development like myself, we all want to see a cure. And we're still working on that. And I think we've got some more work to do on that. But can we get as close to that cure as possible with our existing technologies? I think we've taken a huge step forward with some of the recent scientific advances. So it's not perfect, but I think compared to where we are as a field 5-10 years ago, I think that there's been some tremendous advances.
I want to follow up with you on that new drug development piece of things in a moment. But actually first want to make sure, I don't know, you may have mentioned this. If so, let's underscore it. If not, I think it's important to mention. But Qfitlia also doesn't require cold storage. So when we consider transportation and logistics, right, there's some notable characteristics there.
Yeah, because it's a RNAi or siRNA technology, it's very short. It's only 23 oligotides long, very short.
Oh, oligotides. Yeah, we're all familiar with oligotides, obviously. Yeah.
That hung me up a little bit too. Even though
I still don't know what I'm saying.
I've said it I don't know how many times. It's very stable. So it comes in a pre-filled pen, and it can be stored on the shelf for X number of months, and can be put in somebody's backpack if they wanted. You wouldn't necessarily need to, because you're only dosing every other month, but very stable in that regard. And so the one thing I just realized that we haven't talked about with Qfitlia as well is, because of this siRNA technology, the knockdown is very stable. So unlike factor replacement therapy, where you have a peak and a trough, Qfitlia is very stable. And because we're targeting antithrombin, it can be used for all people living with hemophilia, whether it's hemophilia A or B, inhibitor or non-inhibitor. Antithrombin is one of these anticoagulants that works late in the coagulation pathway. And so it kind of goes around factor VIII and factor IX, where they typically tend to work in the coagulation system. So some other really unique differentiating characteristics of Qfitlia that patients certainly in the community seems really excited about.
Well, having lived with an inhibitor for 7 years and knowing people who have hemophilia B and an inhibitor, this just presents an option unlike any for that population in particular. So that's pretty exciting. And that's kind of along the lines of what I want to ask you next as we start to wrap up. Let's pretend I am a patient living with hemophilia. Let's pretend, role play with me here, Craig. We're pretending
Absolutely.
I am Patrick, but let's say, I'm a 30-year-old guy. I'm going to age myself down. I'm a 30-year-old guy who's on a standard half-life therapy. And I'm finally ready for a change. The first generation EHLs weren't for me. Didn't really pay much attention to the next generation of EHLs. Mimetics, Hemlibra came out. I started hearing things, okay, but not yet compelled. But now there's enough new things out there. I want you to speak to me first from an Altuviiio point of view and then from a Qfitlia point of view and
Yeah.
make the case for each. Why might I, as this 30-year-old guy living with hemophilia, doing well but not fully satisfied with my treatment, breakthrough bleeds come in, lifestyle adjustments come in, it's not quite right and I know that. So
Yeah.
make the case. Why Altuviiio? What's beneficial about it?
Well, with Altuviiio, I think the levels of protection and the ability to be normal to near normal for a majority of the week are clearly an advantage over all other therapies. And so, if that's what you're looking for, IV therapy, replace what's missing, that is comfortable for you, and you want that protection level that is in the normal to near normal range for 4 days, still in the mild hemophilia range on day 7, then Altuviiio is for you. And you have access to a fridge. It should be stored in a refrigerator, and you should feel comfortable doing IV injections. It sounds like that's what you've been doing on a standard half-life, so that is not anything new.
No, and some of those advantages are quite clear, but let's say I am indeed sick and tired of the IV injections. And Craig, I will tell you, I have reached these points in my lived experience where it's
Yeah.
just I can't look at this anymore. I have to go take a break. I'll come back to this in 30 minutes.
Well,
It's a real thing, and that kind of burnout's real. So
yeah,
for that person, for that individual, for younger 30-year-old,
yeah.
30 pounds lighter me who's considering, okay, what are my options now in the non-IV administration? I'm thinking about the mimetics. I'm thinking about Qfitlia. Make the case for Qfitlia.
Yeah, well, Qfitlia is going to provide you very stable protection levels with a small volume subcutaneous injection every other month. And you're going to be able to measure your protection level. Very stable, won't really fluctuate. Your protection levels are going to be nice and even. There's no risk for inhibitor development with Qfitlia. And, because of that, you really kind of open yourself up to not needing to be always next to a refrigerator, etc. So I think both are obviously tremendous options for people living with hemophilia. And I think that is one of the reasons at Sanofi we were really excited to bring all of these new therapies forward to complement Alprolix, which is also available for people with hemophilia B. That options are better than no options. And these give people living with hemophilia, I think, some pretty amazing options now.
Well, I agree. And again, I learned from speaking with you prior to today. And I thought by coming on today and spending this 30 minutes or so together, we'd help some people understand a bit more about Qfitlia in particular, but hopefully about Altuviiio as well, because I, again, don't want to take for granted it's still relatively new and
Yeah.
not as well known by a lot of people. Is there anything, Craig, before we wrap up that we haven't touched on about either Qfitlia? Well, actually, I did want to ask you about this, so never
Yeah,
mind. I'm retracting my open-ended question for something more narrowly scoped.
sure.
Drug development. You mentioned being in new drug development. I'm curious about what's currently in the clinical trial program that either you're working on most prominently or is top of mind as I ask this question. What's in the pipeline?
Yeah, at Sanofi, we are pretty active right now in terms of looking at the community and seeing where the next big step for hemophilia will be. Obviously, I think gene therapy has kind of had a little bit of a peak, and it's at a little bit of a trough now.
I see what you did there.
But I think that we are, obviously supporting as best we can the launch of these two new drugs right now. That's our immediate focus. But we are obviously, is very committed to rare diseases, also looking at, okay, well, when we think about next steps with, gene therapy, et cetera, where is there an opportunity? And so that's about all I can say right now. There on next steps at Sanofi.
Fair enough. Well, that's exciting.
Yeah.
Craig Benson, I appreciate your time and the clarity that you helped provide today. I look forward to hearing feedback from this episode. So certainly if you learned something and would like to give some feedback, please do, because I'd like to have more conversations like the one we just had, as the landscape for hemophilia treatment is more nuanced and complex than ever before. These kind of conversations, I think, are more important than perhaps ever before. So Craig, thanks again for your time.
Honored to be on a guest today, Patrick. Thank you so much.
Thank you again to Craig for joining me here on the Bloodstream podcast. So we'll go from novel available therapies now to, I guess gene therapy is also available. Amy, they're just all available now.
They're
Once
all
upon
available.
a time, these things were out in the future, and now they're
I
just getting
know.
approved and approved and approved. So
I know.
on to our latest gene therapy segment.
Well, this gene therapy segment actually lends really well into the previous segment, because it's all about that decision-making process. We're not going to go into the science on this next segment of the gene therapy segment, A Whole New World. We're really going to dive into that psychological battle of, whether or not to pursue, in particular, gene therapy for hemophilia B. This episode really explores on what it feels like to make that decision. What role identity is about, personal goals, and timing, of course, is all a big deal.
Sure.
Hannah has Chip Haley, who lives with hemophilia B, who has walked through this journey on to kind of discuss what his journey was like making that decision, hearing about timing, and really what it's meant for him since, which is fantastic. And we're also going to hear from Simon Fletcher, a research nurse and a PhD candidate, who really breaks things down from a clinical and compassionate lens when it comes to making this decision. So it's a really honest look at all the hopes and the questions and the realities that come with taking this huge next step. So Hannah, on to you.
Welcome back to Gene Therapy, A Whole New World. It's your girl, Hannah, and today we are diving deep. Gene therapy is a big deal in the bleeding disorder space, and we've talked a lot about the science, the safety, and the results. But today we are focusing on something different, the decision to say yes to gene therapy. What does it really take to say yes? What makes someone feel like it's the right time or even the right fit? Well, the answer, it's deeply personal and rarely simple. To help us unpack this, we're joined by two people who bring powerful and complementary perspectives. Chip lives with it. Simon supports those considering it. And together, they help us explore both sides of this decision. Before we dive deep with Simon and Chip, here's a quick word from our sponsor. It's a Whole New World gene therapy segment brought to you by CSLBehring and their hemophilia B gene therapy treatment. To learn more about gene therapy, hear patient stories, and view additional support resources, visit beyondhemb.com or download B Support wherever you get your apps for more information and to manage your treatment journey. Meet Chip. He lives with hemophilia B and didn't wait for change. He moved toward it. His story is one of determination, of being front-footed in the quest for better treatment. Alongside him, we have Simon, a research nurse and PhD candidate from Oxford, who has guided many through the long, thoughtful process of deciding if gene therapy makes sense for them. We know gene therapy doesn't guarantee perfection. And for people like Chip, that's not the goal. It's about the progress, possibility, and a better day-to-day life.
To me, this was the next step. To me, this was a culmination of decades of research of my healthcare journey. I didn't have high expectations. I didn't have low. So what I've gotten has just been great. So that's something that it's got to be discussed, or outcomes. But if I'd had a lesser value, it's still been a great outcome.
Not every outcome will look the same, but when the results make life a little bit easier, a lot more free, That's a win worth celebrating. Simon has guided many people through this journey, and for him, the first question is always about the goals.
Whenever I've spoken to people about gene therapy prior to them having gene therapy, it's always been a conversation about what do you want to do? What do you want from the gene therapy? What do you want to gain? What do you want to do? When do you want to do it? You know, it's a case of looking, what do you want to achieve with your life?
It's a conversation that goes beyond treatment goals. It's about identity, values, and hopes. At the heart of it is one question: what do you want? And that answer depends on where you are in your own journey. For people like Chip, saying yes to gene therapy came after following years of science, volunteering, and staying ready to say yes.
I heard about gene therapy actually 25 years ago. One of the very early researchers actually sent me a letter and asked me to be a part of her research. And because of other circumstances, that did not happen. 10 years ago, in earnest, my hematologist at Vanderbilt called me and said, "You want to be in gene therapy?"
Chip's story has a happy ending, but getting there meant accepting it might not work out. Still, for many like Chip, the hope and the chance are worth it, even with all of the unknowns still today.
There was a couple of experiences that I had that made me wish for gene therapy just right then. Sure, there were what ifs, but it was short lived. Because I've been involved so long and read so much about gene therapy, even though I was a 25th patient in the phase 3, of course, they'd done 13 earlier in that. Great results. And so maybe there might have been some anxiety, but nothing that was, oh, okay.
That readiness, it looks different for everyone. Some folks can't wait to try it.
One thing about gene therapy, it is really so interesting as a medical technology and medical application. I mean, it's just great. It's fantastic. I think it's going to change many people's lives for the better. When you start at 1.7% and are severe, would I've loved to have been just a mild hemophiliac all my life? Absolutely. I think the base that you start at, of course, makes a difference in your lifestyle and how hemophilia affects you. I was severe. I paid for it.
That's the thing about gene therapy. It's not a one-size-fits-all. It's a personal decision, but for many, it's also a very liberating one.
Some people have needle phobias. Now, I don't, but every once in a while you miss a vein and you're going to miss it again, and you know it. Because I have not had to have factor since then on a regular basis or any factor, I have over 280 times now had not had to give myself anything. So that's 280 times that back of this hand right here hasn't been poked.
And maybe even more powerful, that freedom to finally talk about it.
In the past, I did not share my story of hemophilia because of possible discrimination. People not understanding about my condition. I always wanted to carry my load. I didn't want people to feel sorry for me. But now I want everybody to know what a good outcome I've had and what other young men or older men also, what good outcomes that they can have.
Chip's story is powerful, but it's not about perfection. It's about finding the treatment that fits. Gene therapy may not be the answer for everyone, but for some, it's an answer that changes everything.
It's not the answer, but it's an answer. It's a treatment in a group of treatments. And I think everybody needs to look at all of those treatments in the round and say, what is the treatment that is going to make the biggest difference to my life at this point in time and maybe going into the future?
For Chip, it worked, and the impact still unfolding today.
It could not have turned out any better for me. I've had great numbers. I'm in the normal range. I was normal a week after therapy, and had no side effects. I have they're still following me, and could not have been any better.
For many, gene therapy still feels like uncharted territory. The science can seem overwhelming, but the more we talk about it, the clearer it becomes.
The more people hear about gene therapy and hear about the whole process, then it will remove some of the mystique about gene therapy. And I think there probably is a mystique about it. It's very complex, it's very complicated science. But we also need to hear those stories of people who've had gene therapy, who had a good result from it, but maybe the whole process wasn't quite what they thought it might be. So they can tell their story of what that process was, so that people can then turn around and say, okay, well, I know more about the process now, so that if that happens to me, then I'm not surprised by it.
Gene therapy is more than reducing bleeds or raising factor levels. It's about reclaiming your time, your freedom, and your future.
Success is what the person says it is, where the person says it is, when the person says it is. So we can define success however we want. The people that are having the treatment will define it for us, or should define it for us. And I really think that actually, if we want to know what success is, we need to listen to the people that are having the treatment, so that we know what they consider success to be.
But what does that future really look like? A year out, 5 years, 10 years? In our next episode, we're exploring longevity. Not just how long gene therapy can last, but what lasting change truly means for the bleeding disorders community. Because when your treatment shifts, so does everything else. Your goals, your mindset, and your day-to-day life. So stay with us as the journey is just getting started, and trust me, you do not want to miss what's coming next. It's your girl Hannah, and we are signing off with today's Gene Therapy, a Whole New World.
Thanks again, Chip, Simon, and Craig for coming on today's program. Thanks as always to our presenting sponsor, Takeda, bleedingdisorders.com to learn more. Thank you as well to CSLBehring for support of the gene therapy segment. And thank you to our segment sponsor, Genentech. Visit hemashort.com to watch that beautiful short film. We could not produce this show without the support of our sponsors and advertisers, and we are grateful for that support.
Yes, yes.
Bordeaux, next time we talk, it'll be the second half of 2025 somehow. We'll be on the other side of the dividing line. July 11th is the next episode, and what can listeners expect to hear?
Well, we have Matthew Zachary on the program, and Patrick, you spent time with Matthew. Can't wait to hear what you think.
Yeah, he is a prolific podcaster, speaker, advocate. He has a rich history in the cancer space as someone who was diagnosed with cancer as a young adult and found a gap in the cancer world amongst young adults in particular. Older people, kids, young adults. So Zachary has done a lot of work to try to fill that space over the course of his life and career. I'm a big fan of his. We touched on a number of things that he does. It's a pretty packed half hour, and I hope you enjoyed the conversation.
Awesome.
Awesome. So with that, that is all for this episode, but be sure to subscribe to the Bloodstream Podcast wherever you listen to have that next episode delivered to you the moment it goes live.
And listeners, as always, email us at mailbag@bloodstreammedia.com to inquire about casting opportunities, to share your story here on Bloodstream or maybe on some of our video projects. But mailbag@bloodstreammedia.com is that email address, and you can always find us on social.
I am your host, Patrick James Lynch.
And I am your other host, Amy Board.
And until next time, take self-care of yourself. Bye, everybody.
Bye-bye.